Well, hello everyone. Today in this video, we're going to start chapter 3. We're looking at the basics, the composite cell and plasma membrane. So remember, the cells are the basic unit of the body. We went over the idea of the building blocks in chapter 1. We said subatomic particles make atoms, atoms make molecules, molecules make macromolecules, and that's what we studied in chapter 2, molecules and macromolecules. And in this chapter, we're going to look at the cells and their building blocks, which are called organelles. So as an atom is to matter, cells are to life, and cells the basic structure and function of the body. They're very small. It says it's measured in micrometers. Not everybody knows exactly what a micrometer is, but it's a very small. So anyway, I think everybody kind of knows that. Now, cells, as they grow, they will become what we call differentiated. Again, we'll bring this up a couple of times during this chapter, that you start off with an egg and a sperm. That first cell, when it starts dividing, it is generic. We can't tell the difference from the different cells, but fairly shortly the cells will start becoming unique, and you'll see patterns. You'll see cells that will divide and start becoming the nervous system or the muscular system or whatever. And we refer to that as differentiated. That's all it means, is it becomes specialized. All right. Cells vary greatly in size and shape. Again, as it said there on the last slide, remember that the shape and the function are very interrelated, anatomy and physiology. So this is a little example of some of the different types of cells. We have some cells that are really big and some cells that are really small. It is kind of odd that basically the largest cell in the body, or that the body produces, is an egg, an oocyte, and the smallest cell is the sperm, and those are the two that get together to start all of this. But there's a wide range of the way cells look. Now, when we start looking at cells, one of the main things we do is we have this thing called a composite cell. It really is kind of a Franken-cell. It's just when we take all the properties of different cells and we put them into one cell so we can study them. And we call that a composite cell. I hope that nobody has a cell that's going to look like this composite cell, and if you do, you really need to go get checked out. But generally, cells are going to have three main things: a nucleus, cytoplasm, and a cell membrane. Now, first of all, understand, not every cell has a nucleus. Red blood cells do not have a nucleus, but most all other cells are going to have one. Another thing is cytoplasm. Most people come into this class having learned, for whatever reason, that cytoplasm is the liquid inside a cell, and that is not true. The liquid is called cytosol. What cytoplasm is the cytosol, the liquid, plus all the organelles that are in there. So cytoplasm is more than just the liquid. All right, so cytosol is specifically the liquid, cytoplasm is that liquid plus the organelles. And then we have the cell membrane, and we're going to look at the cell membrane in this video. So this is what we said was a composite cell. Like I said, no cell in the body should look like this. It is just taking all the different parts and putting them in. So we'll see things later on through these videos in this chapter. We're going to see things like the mitochondria. Most people know the mitochondria is the powerhouse of the cell. We're going to see things like the rough and smooth endoplasmic reticulum. We're going to look at this really amazing structure called the Golgi apparatus. Sometimes you might have learned it as the Golgi bodies. So we're going to look at some of these organelles. That's what we, again, in that stepwise process of building the body: subatomic particles, atoms, molecules, macromolecules. The macromolecules were the organic molecules, by the way. And then those macromolecules make organelles. And so those are these in here. Now, the cell membrane is the boundary. All life has to have a boundary between it and the outside world. It's how it is separate. If it didn't have that, it would just disperse. Now, the macro part of it would be our skin. We have a membrane called the cutaneous membrane that is commonly called the skin that protects us from the outside world, and it does a really good job. Now, in a cell, whether it's a single cell organ or just a cell inside our body, we have to have a membrane, something that can regulate a difference between what's on the outside and what's on the inside. And that's what this plasma or cell membrane does. And it is referred to as selectively permeable, which means that it can regulate most things that come and go. But we'll see there's a few things that can slip through. On this membrane, we're going to have special things that can pass signals and respond to messages in the body. And we're going to see that this is going to consist mainly of lipids, fats, and proteins. There's going to be some carbohydrates in there that will help connect and communicate, but it's mainly made up. We're going to see this phospholipid type of a cell or a molecule that makes it up. And that's what we talk about here. It's called a phospholipid bilayer. We'll see this in the next slide, picture of it. But the way it works is each one of these phospholipids is going to have this head and then these little tails that stick down. And what's going to happen is on the opposite side, I'm going to have a head and then these tails that stick in the middle. And so I'm going to have an outside layer of these heads as well as an inside layer of these heads. And the middle layer is going to be all of these little tails that stick in between it. Now the heads are what are referred as water soluble. They are, again, scientifically called hydrophilic. Philic means loving. Hydrophilic means water loving. Like Philadelphia is the city of brotherly love. And if you've ever seen a sporting event in Philadelphia, you might think that it's not the best name for it. But anyway, Philly means love. So hydrophilic means it's water loving. Now in the last chapter, in chapter 2, when we were talking about those covalent bonds, I told you that those polar covalent bonds are water soluble. All things that are water soluble are going to be, as far as the large molecules go, are going to be polar covalent bonds. Then the middle, these little tails here in the middle, these are called water insoluble or fat soluble, lipid soluble. And they describe them here as hydrophobic. I think everyone knows phobic means scared of. It doesn't like water. It's water hating. And these are nonpolar covalent bonds. Again, we looked at that in the last chapter. So the way this works is these little polar heads, which have little charges to them, are going to attract things to it. Now that's on the inside. I should have put it here since I did just a minute ago say that these are on the outer and inner side. So it's going to bring things close to it. But because I have this fat soluble middle, it repels it. The polar things can't go through it. And so we're going to see how our body develops channels and pores that allow things that we want to go through. But fat soluble things, lipid soluble substances, since they are not charged, can slip right through, and they will always move in a concentration gradient because they can move through the cell membrane like it is not there. And so that's why it is selectively permeable. Again, as it says, this is going to have lipid-soluble substances that are able to move through it, but not water-soluble substances. And it all goes back to understanding the idea of how those charges work. Anyway, cholesterol is going to be involved in it. It's going to help stabilize it. And these things we're going to look at, we're going to see things that are inside this membrane that can move around. So if a cell is being attacked, so if I've got a cell membrane and something's attacking it here, or if there's something going on here and I have some kind of marker or some protein that needs to go, it can move to get to this spot. And we call that a fluid mosaic model. It's kind of like it's moving like ships on the sea. And so these proteins are going to have many functions, as it says. Some are pores and kind of channels that allow things to go through. Some are going to be receptors. So when we have hormones, for instance, or neurotransmitters, they can bind to it as a receptor. There's going to be enzymes on it that'll help things happen. And then they'll have things that connect and contact and communicate with other cells. And they're also going to be there for identification. They're kind of like your little name tag, so that your immune system, the immunity as it's going through, it can tell that it's you. We're also going to look briefly at these things called CAMs, which are cell adhesion molecules, and it's mainly for what we're going to see is going to be mainly for white blood cells, showing how they can find where they need to go and move. And then there's also carbohydrates. Again, these are going to also be for cell recognition, interaction, and self markers, like I said. Not a big deal on this. I just want you to know that the cell membrane is going to have lots of things on it with lots of different jobs. Now this is a clean version of it. There's nothing studded in it. The next picture or so we'll see that it's going to have it. But this is what we mean by a phospholipid bilayer. So this is going to be inside the cell, intracellular. This is going to be outside the cell. And this little layer is dividing things. And it's kind of, I hate to say it like this, but it's kind of like a fat sandwich. I've got this little layer of fat, this hydrophobic, or said another way, lipid soluble area here that is non-charged. And then I'm going to have this layer of polar heads, which are hydrophilic, water loving, and it's going to have charges to it. So it will pull things close, and then it can decide whether it lets it go. Anything that's charged, for instance. But things that are lipid soluble or non-charged can move right through like it is not there one way or the other. It's going to move in accordance to the concentration gradient. Now this is kind of showing some of the things that are in there. We're going to look at this later on in this chapter about how things can move in and out of cell. We're going to see these little channels. I am not worried about this part, but I'm going to just point out that there are some things like this one, it's called an integral protein. It does not go all the way across the membrane, where this one and this one here does. So this one is called a transmembrane protein. Now they're going to have special names in it, whatever, but again, not worried about it. All I'm going to worry about right now, and it's going to come later, is this little thing called a channel protein. All right. Now, this goes over some of the things that are on there. I'm not, again, I'm hoping you know about enzymes. Again, we'll discuss what enzymes do even more, but we looked at it last chapter. And so the big one is going to end up being these pores and channels later on in this chapter, as we see how things are allowed to move back and forth through a cell, why it's selectively permeable. Now, I just left this in to kind of point out in our class, these clinical applications, if I do not specifically bring them up in class, they're good information, but I am not worried about us going over these, and it's not going to be on the test. So cellular adhesion molecules, this is the kind of the last thing we'll talk about with this, and the cellular adhesion molecules are what help guide cells to where they need to go. And so on the surface of the cell, and on this again we'll talk about white blood cells, we have something called selectins. Selectins, they're on the white blood cell, and as they're moving through, we're going to have little things on the capillary walls that these will bind to or rub against to provide friction to slow the motion of the white blood cell down. And then we're going to have special things called integrins, which are going to bind to adhesion receptor proteins that are going to help, again, guide the cell out. And then the integrins themselves are what are going to direct the cells on getting through the capillary walls towards the infected site. Now, again, there are other of these, but we're not going to go over them. So this is a white blood cell here. So again, we got the little white blood cell here. On the surface, we have these selectins, and you can see they're all over it. So we got them here and here and here and here. Along the cell wall, or the capillary cell wall, we're going to have little carbohydrates on there. And some of them are going to be adhesion receptor proteins, and they're going to bind with the integrins that are on the cell surface. These selectins will slow it down. These will cause it to stop, and then be guided out to go fight the bad guy. And so it's pretty amazing how all this works. Now, as far as the test goes for my class, our first test is going to be on the first 4 chapters. There's a lot of material. This is not going to be on it for this class. We'll talk about it more when you get into 211 and get into the circulatory system and see how this works in the white blood cells. But as of right now, I just wanted to go over it, but this is not part of the test. And that concludes this first video.